So welcome everybody to a new and definitely exciting edition of our webinar on emerging topics in biomolecular magnetic resonance. So we will, upon the consent of the speakers, record the lectures and make them available on the YouTube channel. To remind everybody, please use the Q&A box or raise your hand and then we can unmute you. And we really invite everybody also to stay to the unofficial part afterwards to have further discussions, and you can also put your questions directly to the speakers if there are any left. Again, as every week, there's the early career researcher edition of the webinar supported by the ISMRBS. So please tune in there. Without further ado, I then hand over the stage. The second presentation will be given by James Chou from the Harvard Medical School in Boston. So James obtained a bachelor in physics from the University of Michigan, Ann Arbor, and then actually moved on to do a PhD with Gerhard Wagner at Harvard University, and working already on very exciting structures of proteins involved in apoptotic signaling. And then in 1999, he moved to Ed Beck's lab for a postdoc. Actually then he directly returned to Harvard Medical School and rose up through the ranks from assistant professor and became in 2012 then a full professor at Harvard Medical School. James received a number of fellowships over the years, which to me is really no surprise given his outstanding work over many, many years. So I actually met James on my first day, I think, of my postdoc, because we basically arrived on the same day in Ed's lab in 1999, and we, I think, almost also left on the same day, I guess, or a very similar time. And I still remember sort of our lunch together with Frank and the other people in the lab going out for having sushi and so on. So it was a very nice time. So yeah, James does really excellent work over many, many years now in different systems. So we look forward to your presentation, James.
Okay, thank you. Thank you, Marcus, for the kind introduction. I do also really miss the good old times at NIH Building 5. So, it's great to be here. What I'm going to talk about today is our study to fill the structure gap of the HIV spike protein that we all know in the membrane region. And we have been working on this project for over 5 years now, and very recently obtained a somewhat complete view of the membrane region of this envelope protein. We all know that the spike protein of HIV is really the primary target for vaccine development because it is really the only antigen on the variant surface. It is also one of the most studied molecules in biomedical science, perhaps, especially in structural biology. So the past 2 decades of studies have already generated a vast amount of structural information for the ectodomain region of the spike protein. Let me just use the laser pointer here. But these studies have yet been able to reveal the membrane region of this protein. So what I'm showing you here is a low-resolution cryo-electron tomography structure of the spike protein on the real virus, obtained in 2008 by the Subramanian group at NIH. It basically shows an overall shape of this trimeric complex in the pre-fusion state. In addition, we also have many high-resolution structures of the soluble ectodomain of the protein. This one shown here in yellow is the crystal structure from Pen Brockman's lab, but you can see the membrane region is entirely missing. Okay, so the spike protein is also generally known as the viral fusion protein. It consists of 2 parts. We have the receptor binding part GP120 and the membrane fusion part GP41. The membrane-related components that I'm going to talk about are all in the GP41, so which contains the highly conserved emperor region, which stands for the membrane proximal external region, the transmembrane domain TMD, and the unusually long cytoplasmic tail CT. So why is the membrane region important? Well, obviously, it is important for the fusion mechanism. But another practical reason is that it is believed to be important for the overall antigenicity of the envelope protein. So a compelling evidence obtained by our collaborator Bing Chen at the Boston Children's Hospital is that they found that deletion or systematic deletion of the cytoplasmic tail can lead to a completely different antibody binding profile of the ectodomain on the opposite side of the membrane, which suggests some kind of structural coupling between the ectodomain and the cytoplasmic tail, possibly through the TM and emperor. So let me elaborate further on this unusual finding. So what they found is that incremental deletion of the cytoplasmic tail leads to decreased binding to the trimer-specific neutralizing antibodies, such as the PG16 and the PGT128. Now, this antibody bind at the apex of the ectodomain trimer shown here, and they only bind to the closed conformation, not to the open conformation. So the table on the right shows that you can see the systematic truncation of the CT leads to decreased binding to the trimer-specific antibodies highlighted in green, but at the same time, increased sensitivity or binding to the monomer-specific antibodies highlighted in yellow. So this suggests that deleting the cytoplasmic tail somehow caused the ectodomain to open up, dissolving the trimer-specific binding site, while at the same time exposing some of the concealed epitopes in the closed state. So this is very interesting, and this is also the major motivation for this project. Okay, so as such, we took on the challenge of using solution NMR to tackle the membrane-related components of GP41. As you can see here, the entire membrane region is really too large for NMR. It has 200 amino acids, it's possibly trimeric, and plus the large membrane mimetic system. So it is really too large. So what we had to do is to take an incremental approach that involves piecing together different fragments of the GP41 with overlapping regions. And unfortunately, the overlapping transmembrane region, as you'll see later, in this construct all have essentially the same structure, and which allows us to actually piece together and build a model of the entire membrane region from the smaller fragment. Okay, so for this, we developed a, I would say, rather general protocol for structure determination of TM oligomers in bicells. And it includes 4 steps. The first step is protein reconstitution in bicells with Q between 0.5 and 0.6. So these are actually quite large bicells for solution NMR. And the second step is determining the oligomeric state of the protein in the bicell, which is actually not a drill task. And for this, we use a method we developed, which we call oligomer label or OG label. So in this method, instead of directly cross-linking the transmembrane protein, which is difficult, we non-covalently label each monomer with a small soluble protein. And then we perform cross-linking on the small soluble protein, which is a lot more robust. The third step is basically detecting interchain NOEs and solving the structure. And the final step is determining the membrane partition of the protein in bicelles using a PRE method. So I'm going to come back to these steps later in the context of a real example. But for the time being, I want to mention that this protocol is really the culmination of a decade-long effort from many lab members. But very recently, 3 people in my lab have really made it streamlined for general application, and they are Bill Fu, Alessandro Pia, and Wen Chen. Okay, so what we did is we reconstituted the 4 different GP41 fragments in bicelles with Q of 0.5 and obtained quite workable NMR spectrum. As you can see, the NMR peaks corresponding to the transmembrane core, here shown in red, are essentially the same between the 2 constructs, Emperor TMD and the TMD, suggesting that the structure is essentially the same. And then this shows we also reconstitute other fragments, which includes the TM and the different CT fragments in the same bicell. And this also shows that the TM core region, the peaks are more or less the same. Okay, so the structures are more or less the same for all those 4 fragments. So this spectral comparison suggests that we can probably use the overlapping structure of the TM core to build the structure and model of the entire membrane region containing the Emperor, the TMD, and the CT. This slide just shows that the determination of the oligomeric state of the protein in the bicell. And here the example is on a fragment that contains the TMD and the LLP2 region of the cytoplasmic tail. So again, in the OG label method, instead of cross-linking the hydrophobic transmembrane protein, we non-covalently label each protomer with a soluble GB1 and then perform cross-linking on the GB1, which is further analyzed by SDS page. As you can see here, when you have GB1 alone, even at very high cross-linker concentration, it can only be cross-linked to a very weak dimer band. But in the presence of the bicell sample, you can see a strong trimer band, which indicate that the protein forms a home trimer in bicell. So another critical experiment is detecting interchain NOEs, and this was done by mixing 2 different isotope labeled chains at 1-to-1 ratio. So one chain is N15 labeled and deuterated. The other chain is C13 labeled. We find this type of labeling scheme offers the greatest flexibility when designing experiments to exclusively detect interchain NOE. So for example, here in this case, we used the proton-carbon J evolution to flip the sign of the NOE such that the interchain NOE with J evolution of 0 and 8 milliseconds will have the opposite sign. So subtracting the 2 spectrum will give you the interchain NOE only. Okay, so using this structure determination protocol, we determined the structure of the 4 fragments in the same bicell under the same condition. So this is the structure of the transmembrane domain, which forms a specific trimer in bicell. Note the peculiar feature of a middle arginine in the middle of the TM. We later actually found that this arginine can actually be hydrated via the hydrophilic pore in the C-terminal half of the trimer. So this is pretty interesting. Moving forward, this is the trimeric TMD extending from the N-terminus to cover the conserved ampere region. The ampere in this context also form trimeric cluster and is largely outside of the membrane. So here I want to also take the opportunity to mention that although the ampere structure shown here looks like a very well-defined structure, but our NMR data really indicates that it is substantially more dynamic than the transmembrane domain. So the way I look at it is that the ampere is actually a flexible joint between the active domain and the membrane anchor. So it should take multiple conformations during the process of membrane fusion. So if you're interested in the polymorphism of the ampere, I recommend reading early papers by Gerhard Wagner, then later Mei-Han, and more recently by the Beck's group. Each of those studies provide different structure aspects of the ampere. So let me move on to the next fragment, and this one includes basically C-terminal extension to cover the LLP2 segment of the CT. And interestingly, the CT also has a pretty unusual structure that it wraps around the TMD trimer to form a base plate-like structure. And finally, this is the, okay. And finally, the C-terminal extension to cover the full-length CT, which basically expand the base plate further. As you can imagine from looking at this base plate structure, it is actually hard to solve this type of structure with NOEs only because of the global and long range, position between the TM and the wide base plate. So we really needed plane restraint to solve this type of structure. And we got them by performing an analysis called the Paramagnetic probe titration. So, which we call the Ppt titration. So the Ppt analysis is actually similar to solvent PRE, but with two differences. One is that it is customized to probe the protein depth immersion in the bicell. And this is actually possible due to the use of sufficiently wide bicell, as in our case, such that the solvent PRE along the bilayer normal will dominate, thus provide a reliable position along the bicell normal. Another difference is that we perform titration of the paramagnetic probe as opposed to using just a single dose. And this is really to get a dose response curve so that we can derive the PRE amplitude by fitting for each of the residues by fitting the exponential decay as shown here. So we find that the PRE amplitude is much more sensitive in probing the position of the residue along the bilayer normal than a single dose PRE. And that's why we do this kind of analysis. For example, for the water-soluble GD DOTA probe, which you just heard from Enrica, we find you can see very nice symmetric sigmoidal profile of the PRE amplitude for the transmembrane domain. But for the CT, much of the residue of the CT, they have a very high PRE amplitude, which is consistent of it being at the edge of the bicell, right? And in also too, we can complement the GD DOTA data by titrating the system with a lipophilic probe. In this case, is 16 DSA, and this gives a nice reciprocal profile. So, because we know the transmembrane domain structure already, right? So we can actually use this structure as an internal calibration to derive the position of the CT along the bicell normal. So this is the, shown here is a parameterized sigmoidal function obtained by fitting the PRE amplitude to the TMD structure. And then the sigmoidal function is then used to derive or assign plane restraints for the CT residues. And this plane restraints for the CT residues really helped a lot during explore calculation. So having the NMR model of the entire membrane region, we could then put together a model for the entire spike protein. Very recently, after our NMR structure was published, a group at the Yale reported a higher resolution cryo-ET structure of the spike protein on the virus surface. This time, the resolution is much higher at 11 angstrom, and they can actually assign density for the ampere. So what we did is we fitted our ampere structure to this ampere EM density and derived the model of the entire spike. And on the right, I also want to show another interesting feature of the CT base plate, that there are actually several distinct hydrophobic and hydrophilic clusters that appear to hold the base plate together. And this polar or apolar nature of this cluster is actually highly conserved. So I'll come back to this later, that these interactions actually influence the antigenicity of the entire envelope protein. Okay, so the new structure did raise two important questions. One is, what is the implication for immunogen design for B-cell-based vaccine development, which is something that everyone is interested in. Another question relates to the large CT base plate. The fact that it resides in the head group region of the lipid means severe lipid exclusion in this region. So how can the membrane actually accommodate this type of architecture? So that's an interesting question. So let me first talk about the impact on immunogen design. So we first made several mutations to the ampere region to see the effect on antibody binding. And here we tested a panel of well-known antibody that targets the ectodomain only, okay, not the membrane region, just the ectodomain. We find that the mutating any single residues important for the ampere structure can all drastically alter the antibody binding profile, especially those of the trimer-specific antibodies shown by the red curves here. And then we introduced the mutations that disrupt the interaction between the transmembrane domain and the CT base plate, and look at the ectodomain sensitivity to the same panel of antibodies, okay, ectodomain antibodies, and found that in this case, mutations in this case on the TM side, which disrupt those clusters, can drastically also reduce the binding of trimer-specific antibodies. And the same is true when we mutate residues on the CT side, although the effect is somewhat less. And finally, for the two mutants that disrupt the TM and the CT interaction, we looked at the effect on the antibody binding to the ampere-specific antibody. In this case, is the antibody called 4E10. So this particular antibody cannot access the ampere in the closed conformation of the spike. It can only bind the open conformation. So the interesting result here is that both mutants show about 10-fold increase in sensitivity to this antibody, suggesting that destabilizing the CT base plate can somehow induce a more open state of the ampere as well. So this mutagenesis studies together suggest a unique opportunity to modulate the degree of and mode of ectodomain opening by simply mutating, messing around with the membrane region, which this means it does not touch the ectodomain. And then we can use this to screen for ones that can induce neutralizing antibodies. So this is certainly a new strategy for immunogen design, and we are working with our vaccine collaborators to further explore this idea. So then what is the mechanism of the cross-membrane coupling? Unfortunately, I don't have the exact answer right now. But this is just a crude model for explaining our observations. We believe the TM helix trimer is strongly held together in the membrane, but also dynamic in the way that it is subject to a pretty significant pivotal motion or scissor-like motion. So in the pre-fusion state, the base plate would stabilize this TMD trimer, but when you remove the base plate, the TMD trimer undergo substantial pivotal motion, which can destabilize the anchor, which can further propagate to the ectodomain. Of course, this model still needs to be rigorously tested. However, we do have very limited evidence of the scissor-like motion of the transmembrane trimer. Here, what we did is we used the Emperor TMD sample, and we measured the R1-R2, NMR R1-R2 relaxation rate product as probe for relative exchange dynamics. So we first measured the R1-R2 product for the regular Emperor TMD, as shown with the dark dots here, right? You can already see that there's a substantially higher dynamics for the Emperor and the C-terminal end of the transmembrane helix. And then based on our NMR structure of the Emperor, we designed or engineered disulfides so to lock the Emperor so that it forms a circular crosslink trimer. And as you can see here, locking the Emperor significantly reduced the exchange dynamics of the Emperor region, which is expected. But what's interesting is that it is also significantly reduced dynamics all the way at the C-terminal end, about 40 residues away. So there is some evidence of this kind of scissor motion for the transmembrane helix. Okay, so finally, let me just say something about lipid exclusion by the CT base plate. We know the CT base plate resides in the head group region here of the bilayer. And if we build lipids around this protein, there is a big void around the transmembrane domain, which of course is impossible. So in collaboration with M. Brown of Virginia Tech, we performed all atom MD simulation of the NMR structure in HIV membrane and found that in the first couple hundred nanoseconds of simulation, the LLP2 region of the base plate show ascending movement to fill the void, while also causing some local sinking of the exofacial leaflet. So this is kind of interesting because this means that the amphipathic base plate itself is really filling in for the void to fill the lipid for the lipid exclusion, and perhaps directly interact with the exofacial leaflet. And this sort of makes sense because this can be done without drastically changing the membrane morphology to accommodate this kind of wide base plate structure. So to independently examine this kind of phenomena, we performed the lipid NOE measurement, and we indeed found that the C-terminal half of the transmembrane domain show far less lipid NOE than the N-terminal half. And that this is in strong agreement with the simulation result. So at the moment, I still don't know the functional implication of the CT base plate. The current structure only provides a framework for the many follow-up studies, such as the effect on membrane fusion and even possibly on the interaction with the matrix protein. So this is where we are now. I just want to conclude by thanking the people in my lab who contributed to this project: Alessandro Pia, who's back to Italy now, Bill Fu, Jyoti Dev, and Wayne Chen. Also want to thank our collaborator, Bing Chen and Steve Harrison, who work on the cryo-EM study of the spike, for Mike Seaman for conducting all the antibody neutralization assay, and Anne Brown for the MD simulation. And also want to acknowledge the great facility at the MIT Harvard Center for Magnetic Resonance, and also Dr. Liu for helping with some of the data collection in the facility in Shanghai. So, so I'm done here. Thank you for your attention. I'm ready to take questions, if any.
Thank you, James, for an exciting presentation. So there are a few questions already. I start with the question by Frank Laukien, who asks, can we measure the effects of viral envelope glycosylation on these structures? Are they expected to important, e.g. in immune evasion? I guess that's related to mass spec analysis of these proteins.
Yeah, this is actually a very good question. Yeah, the HIV spike is actually one of the challenge of vaccine development is that the spike is heavily glycosylated. So that's how the virus evade immunity, right? So of course, with the NMR study, we only will focus on the membrane region, so we didn't touch upon the glycosylation of the ectodomain. But our collaborators know about this, and this is in fact one of the challenges, and that's why we believe one possible strategy, a novel strategy, is that if the membrane region, if messing around with the membrane region can actually modulate the opening of this highly glycosylated ectodomain, this may present new opportunities, to expose certain epitopes that are more or less conserved to induce broadly neutralizing antibodies. Did I answer the question?
Yeah, I guess so, but if not, Frank just can hang on and we can discuss this afterwards in the informal part. So then there's a sort of a highly ranked question by Paul Schander, and I guess this maybe is also something which we might discuss in the informal session, but I still read it out because it was raised there. So basically, it's about the differences between the oligomerization state of the transmembrane region in the various studies. So Paul writes, there's a study by Giglieri and Bex that shows under the same condition that the TM helix is straight and monomeric. How can one explain these differences? And could one get additional evidence for the trimerization beyond the cross-linking?
Yeah, so that's also a good question, and we've been dealing with this controversy for quite some time. I looked at the paper, the Jack's paper, very, very carefully, and look at all the details. Indeed, membrane protein sample preparation is tricky and it's finicky, depending on the condition of presenting preparation, reconstitution. It may give different oligomerization state. And from biological, functional perspective, we can even, it is also possible, during the process of fusion, at some point, the transmembrane domain trimer may come apart, right? in fact, most people think it has to come apart at some point during the process of fusion. But there are other evidences definitely supporting the oligomerization state of the trimer. For instance, there is a study by Mei Hong, who's in the panel here. They use solid state NMR to show that it is indeed oligomeric, or if the evidence for trimerization is not very rigorous, at least it is oligomeric. And we have also performed mixed PRE experiments, and that's very robust to show that it is not dimeric in the bicell. And this experiment involves actually the mixing of N15, NMR visible chain, mixed with a NMR invisible chain, but labeled with MTSL. In this case, we can clearly see very specific broadening or PRE effect that's consistent with a trimer structure. So that's my short or long answer.
Thank you. I think, Paul can definitely stay on and add and so on, and we can discuss this, I think, in detail further on. So then Mei Hong would like to know what constraints did you obtain for the turn residues in the EMPA region? And secondly, is the N-terminal half of EMPA...
Sorry, Marcus, can you repeat the Mei Hong's first question again?
What constraints did you use or obtain for the turn residues in EMPA? That's the first one. And the second one is again similar to the EMPA, but now asking which restraints, PREs, or whatever you have for defining whether the N-terminal or the C-terminal half is closer to the membrane.
Okay, let me first address the first question. And those are intermolecular NOEs. Okay, so we see quite few intermolecular, unambiguous intermolecular NOEs between the different chains, between EMPA from the different chains. So that's how it was determined. So it was basically NOEs.
And this includes the turn residues, basically. I guess that's what
this includes
I would like to know.
A kink. You turn residue, you mean the elbow here, right?
I don't know, but I guess.
Yeah. So yeah, that's in fact, this is consistent with the monomeric structure that the report published by Gerhard Wagner in detergent micelle. They also see this kind of a kink and a turn. So this is consistent in the context of this particular sample, because the TM is trimeric, and this probably promoted the trimerization or clustering of the EMPA. And we do clearly see NOEs for that. What was the second question again?
So regarding sort of whether the N-terminal or the C-terminal part of EMPA is closer to the membrane, and if you have any PRE data or other data to restrain this, basically.
Yes, I didn't include a slide to show this solvent PRE analysis. So we basically performed both GD DOTA, solvent PRE, and the lipophilic probe solvent PRE or lipid PRE. So majority of the EMPA region shown in cyan here are actually outside of the membrane. But parts of it, of course, is right at the edge, but the majority actually extends out. And we also have independent data that antibody can actually bind to the EMPA in the context of our bicell sample. So majority of the N-terminal, which we call this N helix of the EMPA, is actually exposed. And this is actually good news for antibody development, because EMPA is perhaps the most conserved region of the spike. So any antibody against EMPA would automatically be broadly neutralizing. But so far, challenge is still how to present the EMPA to teach the immune system to generate those antibodies.
Yeah. So there's actually a question by Anne McDermott. a little bit related to this, and this is to the experimental design of the PRSEs and in particular the gadolinium DOTA. So N writes, although the GD DOTA tech is water soluble, it may be relatively concentrated near the head groups. And isn't there scattering data indicating this? And then the critical question again comes, how would that affect, let's say, then the interpretation in terms of the positioning in the membrane and so on? If you have this concentration of the gadolinium at the head groups.
Right. I mean, it is possible there is a concentration of GD DOTA around the surface of the bicell. Although I think this shouldn't really affect our analysis because we basically perform a dose response, obtain a dose response curve, right? So it doesn't matter how it is concentrated at the surface of the bicell, you still have we are basically, in other words, we're not using this tau here, the decay constant, as a probe for each resin. We're basically fitting to extract the PRE amplitude, overall amplitude. And this is why I think the mechanism or whatever the concentration, local concentration of GD DOTA around the bicell should not fundamentally affect the outcome of our analysis.